in PCR. PCR is the basic technique that we use in laboratories. Almost like in every experiment we require PCR. So PCR stands for polymerase chain reaction, and the instrument that we use to run this reaction is called a thermocycler. Okay. So this PCR is okay. Before we do that, this PCR is nothing but we are trying to make multiple copies of the DNA. Okay. In a normal cell, if you see on the left side of your screen, this is replication, right? In a normal DNA replication, you look at a lot of enzymes. Okay, so that one DNA strand is divided into two, okay, and is equally distributed between the daughter cells. So initially, you require a DNA helicase, okay, that unwinds this double-stranded DNA. Okay, then you have these SSDP or single-stranded DNA binding proteins that bind to this denatured DNA so that they don't re-anneal. Okay. So what happens? Helicase moves towards the coiled DNA, okay, and opens the DNA that's behind it. While moving forward, there's a probability that this DNA strands can again denature. So these single-strand DNA binding proteins come and they bind to this single-strand DNA so that they don't let them to these denatured DNA strands to renature again. So while doing so, another enzyme like RNA primase comes, it joins a primer, okay, in 5' 3' direction, and then DNA polymerase 3, 1 or 1, they come and they start kind of rebuilding or synthesizing a complementary DNA strand, okay, both of these strands. The replication always in 5' 3' direction, as you know. And then once this polymer, can you see my cursor? Are you able to see my cursor?
Yes, sir.
No, sir. No, sir.
Cursor you cannot see?
No, sir.
Now you can see?
Yes, sir.
Yes,
Now
sir.
you can see the cursor? Yes or no?
Yes, sir.
Yes, sir.
So you can see the screen also, right? And you are able to hear me properly, or the voice is breaking?
So the voice is breaking.
So I'll try to record the video also. Okay. So then once this helicase is moving, cursor you are able to see, right? So once this DNA helicase moves towards this end, so there's a supercoiling of this portion of the DNA and the helicase cannot move further. So what happens then? These topoisomerase 1 and 2 come. If you remember the chapter or lecture on linking number, how tits and rits are formed. So when you overwind this DNA due to the opening here, so it overwinds this DNA and there are twists formed in this DNA, rits formed in this DNA. Okay. Now these are relaxed by your topoisomerases, 1 or 2. If there's one nick in one strand, topoisomerase is responsible, and if you have 2 nicks, then topoisomerase is responsible. Topoisomerase 2 nicks, it decreases the linking number by 2, that's why it's called as topoisomerase 2. Topoisomerase decreases linking number by 1, that's why it's called as topoisomerase 1. Okay, and then once these strands are formed, okay, leading strand is good, but in the lagging strand there are discontinuous replication, and you have a primer here. Okay, it synthesizes the DNA like this, and then you have another primer is transferred. Then when this DNA reaches here, there's a process called primer chewing, and this primer is dissolved because it's RNA primer. I don't want to go in detail in replication, but what happens then you have two strands here, and they are separated. So then DNA ligase comes and it joins these two strands on the ligand strand. So there are so many enzymes responsible for DNA replication, but in case of PCR, we just heat and cool it. We only use two things. We heat it and cool it. These two things are used instead of these four enzymes. And only one enzyme that we look at is polymerase or Taq polymerase. That's a thermostable polymerase to run a PCR reaction, right? So the goal of PCR is just to make multiple copies of a DNA. Okay? So if you go to the history of the polymerase, this is the timeline, okay, of different discoveries and inventions in biotechnology, molecular biology. So in 1953, Watson and Crick they gave the double helix structure, and then there was a scientist called Clappy who first showed that the through enzymatic assay, he was able to replicate short DNA strands. Okay. Then in 1976, a polymerase called as Taq polymerase that was shown to be heat stable was isolated from Thermus aquaticus. That's the organism that grows in volcanic vents. The temperature is very high, and still they are able to survive there because the enzymes are stable, heat stable. Okay. So they isolated this Taq polymerase from this Thermus aquaticus. And today also we use this Taq polymerase in our day-to-day PCR reactions. Then in Caramulus developed a PCR. Okay, how he did that? He made different water baths at different temperatures, and he then put the DNA and the primers and the Taq polymerase in those different temperature water baths, and he did like that. But now we have a PCR machine that does the same thing for us, okay, in a shorter span of time, and is less time consuming, plus it's easy, user friendly. So then Caramulus shared the Nobel Prize in chemistry for this PCR. So you can go through these timelines. Okay, now once we run the PCR reaction, so how things work here? So you have two strands of DNA. Okay, you have two strands of DNA here, and you can see you have 5' 3', and these strands run anti-parallel. If you have an A here, you have a T here. If you have a G here, you have a C here, right? So these strands are complementary but run in opposite direction. That's why we call them they are anti-parallel. So what happens in the PCR reaction? First of all, we heat these DNA strands at 92 degrees centigrade. At 92 degrees centigrade, what happens is that these bonds that connect these two strands. So what are the bonds that connect two strands of DNA? Anyone? Anyone? None? So these two bonds, the bonds that connect these two strands of DNA are hydrogen bonds. If you have a G here, you have a C here, then you have a triple hydrogen bond. If you have an A here and a T here, then you have a double hydrogen bond. This you study in 12th class. So what happens? This is called as initial denaturation. So at 92 degrees, these strands separate. Once they separate, then we bring down the temperature to the TM temperature. Okay, we have added primers to this mixture, and you have a forward primer and a reverse primer. For a successful PCR reaction, you need to have both the primers should work. Okay, and the temperature at which these primers bind to the DNA strands is called the annealing temperature. Okay, and when we bring down the temperature, okay, the DNA tends to renature back. So since primer concentration is very high, these primers bind first. Okay, once they bind, then we bring up again the temperature to 72 degrees. Okay, this step may vary in different PCR reactions because you can have different TM for different primers. So we can vary this in a PCR reaction depending upon what gene we are amplifying. Then when we bring up the temperature 72 degrees centigrade, 72 degrees centigrade is the ambient activity. Taq polymerase has the ambient activity at 72 degrees centigrade. So at 72 degrees centigrade, the Taq polymerase starts rebuilding these strands. Okay. So how it rebuilds, we add DNTPs also, we add DCTP, DGTP, GTTP, and GATP. These are the building blocks of a DNA. And once, if you have a T here, okay, then DTTP or the DATP will come here and will form a base here. So on and so forth. This Taq polymerase acts as a polymerase and builds another strand. So at the end of this reaction, this forward primer builds one strand and the reverse primer builds another strand. So at the end of the reaction, you have this double strands of DNA formed from this one single strand. Okay, these are from this strand of DNA. Okay, you have now two copies. From one copy, now you have two copies. Okay, this is one cycle. Now, if you have n cycles, then the formula which you use, that how many copies of DNA you will have after n cycles, is 2 to the power n. Okay, so you can see here it's a graph that shows DNA copies, okay, vis-a-vis the cycle numbers. So if the efficiency of polymerase is 100% and the binding of primers is 100%, then only after n cycles you have 2 to the power n copies of your DNA. So if you run this PCR reaction, that means I'm saying if this from here to here, it is one cycle. After one cycle, one strand of DNA gave rise to 2 to the power n. N is 1 here. So 2 to the power 1 is 2, right? So you have two copies of DNA. So similarly, now if you have n cycles, say 30 cycles, after 30 cycles, you will have around 1 billion copies of DNA. You have almost 1 billion copies of your DNA will be made after n cycles in the PCR reaction. Right? Any questions still here? Any questions still here? I know you people are always so silent. Okay, moving further. Now, why do we need this PCR? Okay, there are various applications of PCR. It can be used in molecular identification. You can use it in, say, molecular identification. Okay, in archaeology, molecular epidemiology, okay, ecology. Then you can use it in DNA fingerprinting. If you want to catch hold of a suspect, okay, involved in a murder, or you want to check the parenting of a kid, like if you remember that tsunami baby. Do you remember the tsunami baby? So there was a baby who was found after the tsunami that took place. Okay, and I think Indonesia or somewhere. So then two parent couples said that this baby is ours. Okay, so what they resorted in a fingerprinting, and from that DNA fingerprint, they came to that two of the parents, the one couple was kind of faking it, but the other couple was real parents. So the baby was handed over to that couple. Also, it can be used to solve murder mysteries if you have some suspects that have kind of done something wrong, have murdered someone, or have kind of involved in a burglary or a theft. So you'll be able to catch hold of those using DNA fingerprint. You can use it in DNA classification of organisms, like we do DNA barcoding. We get different samples, we isolate DNA, then we amplify certain DNA sequences like IPS regions or ribosomal RNA regions or say a MAT-K gene or COX gene. So once we get the sequence of these using PCR, we are able to classify these organisms. Can be using genotyping, like you do RFPD, RFLP, there are various markers that you can use. Okay, you can do a prenatal diagnosis of someone has born error of metabolism or someone is some parents are like want to know the health in general of a baby. Is it okay? Does it have any congenital deformity? Or what you do, you just take the DNA sample from the amniotic fluid of the mother, and then you check for various disorders. You can do mutation screening. You can do drug discovery. You can manipulate using a PCR, and you can also detect pathogens like various PCR-based tests are done to detect various pathogens like these days you have this COVID-19, you do a real-time PCR to check if the pathogen is present or not. So then you can use PCR in sequencing also. You can use it bioinformatic analysis. You can use cloning and was used extensively using Human Genome Project. Okay, then you can use the PCR in genetic engineering, you can gene therapy. The red ones here are the applied research, and the blue ones are the basic research. These are many applications of your PCR. So for molecular identification, where PCR is used, you have an RFID restriction fragment polymorphism. This is not in your course, but if any of the students want to kind of know the mechanism of RFID, I'm glad to share another lecture on that. Okay. You have REP PCR, as repeated to sequence-based PCR, then you have randomly amplified polymorphic DNA, the PMD. Like these days, I've been doing this technique to kind of do genotyping, or it's also a marker that's used. So we use extensively PCR using this. Okay, then you have CFLP, cleavage, fragment length polymorphism, then you have amplified fragment length polymorphism or AFLP, and then you have sequencing. So where these are various molecular identification tools where PCR is used. Okay, in genotyping, for example, one study we were doing is that if you remember, we did one Felox. I have one lecture on YouTube on Felox recombination. If you can go through that, okay, I'll share the link on the Google Classroom. So there we were trying to make these EL2 knockout mice. And how we did that, how we know that this EL2 is knocked out from the mice, we cut their tail and isolate DNA and run a PCR. If the band was hit, we got a single band at this location. Okay, this was showing that the locked sequence is here. Okay, this is not a knockout. If you see a band at this location, so we know that okay, the lock speed has been mutated, because what we do actually induce a neomycin cascade between the lock speed gene that disrupts this gene. So the primer that amplified this sequence earlier are now amplifying the larger sequence that has a fragment inside in between. Okay. So and if you have both band, that means heterozygous and heterozygous. So we can use this PCR in that also. So there are various types of PCRs that we use, and almost we do all of these in our lab. We have a quality PCR, multiplex, FFP PCR, hot start PCR, in situ PCR, inverse PCR, an asymmetric PCR, long PCR, anchor PCR, allele specific PCR. touchdown PCR, PCR. Okay, then you have the assembly PCR, digital PCR, suicide PCR, that's usually used in archaeological studies. You have a raw reverse transcriptase PCR, semiconductor RT-PCR or real-time PCR. Then you have nucleation-specific PCR to determine if the DNA is nucleated or not, and then you have a real-time PCR. Colony PCR, what we do, so usually if we have to kind of detect a particular, say, plasmid or something, what we usually do, or a DNA, which type of DNA is present in that, or which type of organism is that, usually what we do with culture, like what we did in plasmid DNA isolation, we cultured the bacteria first overnight. We took the colony from a petri plate, okay? Then you cultured it. But here we don't need to culture. What we do is simply take a colony, okay, from the plate, okay, then put it in a PCR tube, add primers and water, boil it at 100 degrees centigrade. The cells are disrupted, the DNA comes out, okay, and what happens after that? PCR reaction is done. Once the PCR reaction is done, we'll be able to detect what kind of a pathogen is that. This is called a poly PCR. It's very easy. We usually do it in the lab. If you clone something, you want to see that we have cloned or DNA genotyping. So what we do, we pick these clones from the plate and then kind of run a PCR directly into the DNA. Then you have a nested PCR. Okay, it's a when we want to be more specific about the fragments that we have amplified. So here we use two sets of primers. First set of primers, we amplify this target sequence, plus we amplify the flanking sequence also. When we run a primer, these two primers, always remember that the DNA between these two primers gets amplified in the PCR. So when this much is getting amplified, you can see here, but the target sequence is only this. To be more specific, what we do, then we use another set of primers that lies between the first set of primers. So if the first set of primers were here, the second set of primers will be here. Okay, more specific. So now, if we, in the first instance, we have wrongly amplified or wrong amplified, the DNA region that's amplified is called an amplicon. Now we are using a second set of primers, and if we have amplified a wrong set of sequences in the first instance, these second set of primers will not work because they will not be specific for this sequence. They will be specific for this sequence. And these two primers immediately somewhere else, and they amplified a sequence. But what our sequence was not in that sequence. So to be more sure, what we do, we run nested PCR, and if we run flanking primers, okay, which amplify the target sequence plus flanking regions, then we use primers, okay, that are, okay, targeting the target sequence. Then we amplify it. And when we get this sequence, we know that we have a PCR reaction is successful. If we do not get the sequence that we initially, we have kind of run a wrong set of primers. Then you have multiplex PCR. Now you know you have like hundreds of samples of, say, different strains of bacteria. You want to amplify them. So what you do, you design primers in such a way that if strain A you amplify, you get a fragment, you get a, say, band of, say, 275 base pairs. If you make a primer for strain B in such a way that when the PCR amplicon you run on the gel, you get a band of 141 base pairs, and so on and so forth. So what you can do, you can put all these four in one tube, add these four as a primer, then amplify them. The band that you obtain, okay, here will tell you that which of these is present. If, for example, say the 275 band is not present, then you say that strain A is not present here, but strain B, C, and D is present. So this kind of PCR reaction saves your time, because in one PCR reaction you run many samples. It's called as a multiplex PCR. AFLP PCR or amplified thermal PCR is a marker that we use to distinguish between, say, two organisms. So here what we do, We take the DNA out, then we just digest it. Two sets of enzyme. One is a frequent cutter, one is a rare cutter. A rare cutter is the enzyme that recognizes a larger sequence, say like 6, like GATTC. These are 6 base pairs that are recognized by a rare cutter. So the probability of finding this rare cutter in a sequence will be 4 to the power 6, right? So say after, if you have the restriction site at position 1, then after 4,000 base pair you will have it again. However, a frequent cutter like MSC1 recognizes 4 base pairs. Like your SAW3A1, like GATC, it only recognizes 4 base pairs. So the probability of finding this sequence again in a DNA fragment will be higher, because 4 to the power 4 is equal to 256. So if you find the sequence at position 1, then after 256 it will be again. So a frequent cutter crosses DNA many times, a rare cutter crosses DNA less than times. Cutter DNA with two enzymes, a frequent cutter and a rare cutter. Okay, then we ligate adapters. These adapters are nothing but they are sequence of DNAs, double-stranded DNAs. Okay, and to these adapters, then we add the primers. To win a PCR reaction using forward primer for rare cutter and a reverse primer for a frequent cutter. And only those fragments will be amplified where one end is a rare cutter and other end is a frequent cutter. So I will kind of have one lecture on this on YouTube. So we use PCR here also. Then you have a hot start PCR. Usually, when you have enzymes that are stored in lab conditions, sometimes what happens, these like a tag polymerases in enzyme. So they have this intrinsic activity and they're active. Okay, and this decreases the efficiency of a tag polymerase. So what they do, they add antibodies with this tag polymerase that are specific for this tag polymerase. So once the antibodies are bound, this tag polymerase is inactive. So when you put it on in a PCR and heat it, initially denature it at 95 or 92 degrees centigrade, these antibodies are separated from the tag polymerase, and now this tag polymerase becomes active, and then it will bring about the polymerase chain reaction. So to increase the efficacy of tag polymerase, okay, we use this hot start PCR. Another type of PCR is your in situ PCR. So in situ PCR, what you do, in situ means in cell. Okay, you have that in vivo, like if you do an experiment in mice or a plant, it's in vivo. If you want to do an experiment in cell culture, that's in vitro. And if you want to do an experiment on a slide, it's called in situ, like fluorescence in situ hybridization. Or if you want to do an experiment on a computer, like with bioinformatics, so that's in situ. Okay, so in situ is on the slide. So what you do, you have the cells, you fix them on the slide. Okay, then the add PCR reagents and put in a sealed box, small box, and then there are special boxes for it. And then you run a PCR reaction, and then you have the microscopic visualization of these. So you PCR, you run it on a slide. Then you have inverse PCR. So inverse PCR, you know that initially I told you that if you want to amplify this DNA fragment, you should have a forward primer here at one end and a reverse primer at other end. Okay, but what if you want to know a sequence, but you only know a certain region that's in between the sequence? Now you want to run a PCR from here and here, but you don't know the sequence of the sequence. So what you cut this DNA and then ligate it. Once it is religated or recircularized, you have this known portion and this is unknown portion. Okay. Then you know, since you know the sequence of this known portion, you just digest it. Once you digest it, then this known portion is divided into 2 portions and it's flanking this unknown portion. Now you have a DNA sequence in between, and you can put a forward primer here and a reverse primer here. It will amplify this sequence, and by doing this PCR, you'll be able to know what this region is. This is called an inverse PCR.